This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 657, recorded on August 26th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Howdy, Vincent. We got 84 degrees, headed for 95. We still got another, week and a half at least of temperatures up around 100. And then the phony 2-week forecast suggests that maybe it's going to start to cool off. But we're almost out of this.
Yeah, for you, cool off is like 85, right?
Oh, 95.
95. Okay. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is actually sunny and 71 here. Pretty nice.
From Southeastern Michigan, Kathy Spindler.
Hi, everybody. I'm just checking because the temperature in the thermometer in my window said 68, but the one on my phone says 78.
Wow, it's a big
There's
difference.
Quite a difference between those two. And I think the one in my window is closer to being right. Anyway, one of those would correspond to 25 Celsius, probably the 78 one. But
your video went away. You know that, right?
I know, because I was checking the window.
Okay.
Right.
You remind me of a story late in my mother's life when her neurons weren't totally together, when she called me up one time in the middle of the night and she says, one of my clocks says it's 3 o'clock and the other says it's 3:01. Which is correct? And I said, it's 3 in the morning.
Yep. All right. We have a guest today from a place where I might actually rather be at this point, La Jolla, California. He's at the La Jolla Institute for Immunology. Shane Crotty, welcome to TWiV.
Thanks for having me, Vincent and all. It was 70 degrees on my bike ride this morning, which is warmer than usual.
I have to.
Still nice.
I'm impressed. You got there. It's 8 o'clock your time, and you're there and raring to go. So you're an early guy.
Oh, I get here at 6.
Wow.
Where do you bike from?
It's about a half-hour bike ride from my house.
So up north? I used to live in La Jolla. I was asking specifically, but
apologies. Carmel Valley.
Got it. Okay.
Where did you live, Kathy?
I don't bike past the ocean because it would add 3 extra miles in a giant hill that I just can't quite stomach early in the morning, even though it'd be beautiful to see the ocean.
Wow.
Yeah, no, I lived in Del Mar, La Jolla, and I have some friends who had a nice place in Carmel Valley. So
nice. Yeah, it's pretty nice out there, at least by the coast. Beautiful. I love it. All right, Shane, we're going to talk about immune responses to SARS-CoV-2, but I want to first tell people a little bit about your history. Now, are you a Californian to begin with?
My dad was in the military, so I lived about 11 different places before I made it to college. So yes, but it's also yes for a bunch of places.
Okay.
But I've lived here for 17 years now. It's by far the longest I've ever lived anywhere in my life. So yes, I'm a Californian at this point.
Where did you go to college?
MIT.
Okay. What years were you there?
92 to 95. So, which is where I started coming across David Baltimore, for example, right?
Yeah.
Wow.
Yeah.
He's a youngster. This is, that was yesterday.
Yeah, I was going to.
I'll keep my mouth shut.
I was there from 79 to 82, so not even close to you. And that was undergrad at MIT. Is that right?
Yeah.
And then after that, you went off to do a PhD?
At UCSF with Raul and Dino.
Right. Which is probably where you came on my radar, I guess, since it's a polio lab, and I know Raul well. And then a postdoc was where?
With Rafi Ahmed down at Emory University, studying vaccine immunology, T cells, B cells, all the stuff in between.
Got it.
Really, my PhD was on molecular virology, but there was a vaccine project in Raul's lab that I helped out on. And that was when I saw that, wait a minute, vaccines are just phenomenal medicine. And when we took our data to an immunologist and said, what does it mean? How can we make it better? They said, we don't know. That's vaccinology, not immunology. Nobody knows that stuff. And I was like, wait a minute, this is a big problem. And so that's when I switched directions to try and help fill that gap, which a lot of people have done now in the past.
So wait a minute. I would not have made a distinction between vaccinology and immunology, I don't think. How are they distinguished? Or maybe they aren't anymore.
So now they're a lot more connected. But 20 years ago, it was pretty straightforward to say almost every vaccine that had been developed to date had been developed with minimal knowledge of immunology and certainly minimal knowledge of modern immunology. And there'd been this huge amount of discovery in immunology, certainly from now over the past 30 years. And those generally weren't, those connections were not being made. Those fields basically grew up as separate fields because vaccinology existed way before anybody knew immunology. And somehow they really stayed quite separate, such that vaccinology largely stayed as sort of tried and true approaches and empirical approaches of just, you try things until they work. And you say, well, wait a minute, we know a ton of immunology. Can't we make those connections and turn it into more of an engineering problem? Because that's really always been the big problem of vaccine development, is that if you try one and it works, it's phenomenal, right? I mean, the impacts are just extraordinary, but 99% of the time it fails. And then there's really almost nothing learned from most of those failures in the past. It was just, you went back to square one because the immune responses to that failure weren't particularly interpretable as to why it was a failure. Was your actual fundamental vaccine concept wrong? Or did you just have one piece of the puzzle wrong, and there was one bit of the immune system that wasn't quite being brought in, for example?
So Rich, that was the case when I was in graduate school. And I was sort of a unique person, immunology graduate student in this vaccine lab. It was very strange.
Okay.
So it's gone on for a while.
So, Shane, have been acquainted for some time because you spent some time with Vaccinia. How did that happen?
Yeah, so that was really when I joined Rafi Ahmed's lab as a postdoc and said, look, I'm really obsessed with vaccines and vaccine immunology. What are things we can do? And Rafi agreed that one good path was to study licensed human vaccines, because the immunology of licensed vaccines just wasn't understood, basically, because you have a successful vaccine, it works. Nobody wanted to study it anymore. The problem was solved. And so with something like Vaccinia, it gave us an opportunity to ask, well, there were two things really that we went after. One was, it was a fantastic opportunity to ask about immune memory, because here you've got a vaccine that there was no virus around, right? Smallpox had been eradicated for 30 years. Most people didn't get the vaccine. And so we could go find people who definitely hadn't seen this virus or anything like it in 30 years. Did their immune system still have any memory of it or not? And it eliminated a lot of the problems of trying to understand memory in other contexts. And sure enough, people had pretty fantastic memory, even after like 60 years, from their childhood vaccinations. And so that was a really nice study to be able to do, both in terms of memory B cells and memory T cells. And then the other thing I think that you ended up being more acquainted with that we struggled to understand was when people say, okay, this is a fantastic vaccine. Why is it fantastic. And I think the operating idea of a lot of people was that it's neutralizing antibodies. Those are important. And then it's probably a fantastic antibody response to like one thing. It's like this really great response to the key thing. And our take after a bunch of studies was that, no, that probably wasn't the way it worked. Actually, it was that there were a bunch of neutralizing antibody targets. There were a bunch of good antibody targets, and that the vaccine was so good. It was that in most people, it elicits, let's say, 5 to 7 different good antibody responses. So that there's a lot of backup plans. Basically, it's a vaccine with a lot of safety nets. It's not working for one reason; it's working for a number of reasons. So even people on the outliers of, instead of having 5 good responses, you've got a person with just 1 or 2 good responses. That's still good enough. Is sort of the way we interpret it. So there's a lot to learn from.
Did you study T cell responses as well?
Yes.
And did you get a sense for their role in the success of the smallpox vaccine?
So I'd say the best data on the roles of T cells in the smallpox vaccine come from a human longevity study by Mark Slifka's group that found that the T cell memory did decline over decades, where we found that the B cell memory didn't decline over decades. So there was a difference in the biology there. And then actually a monkey study by Franchini's group, where they could really do a rigorous protection experiment and split up the immunological components. So they did, they basically said, oh, you've really got 3 parts of the adaptive immune system, right? You've got antibodies, helper T cells, and killer T cells. And so what they did was they immunized monkeys with the smallpox vaccine. And as you know, Rich, you can then challenge the monkeys with monkeypox, and that's a good model of the severity of smallpox in monkeys. And the vaccine does work against smallpox. And they could ask, they could have monkeys that they separately tested the role of the antibodies, the helper T cells, and the killer T cells by essentially depleting any of the 3 or transferring them to other monkeys. And what they found was that in that case, it was really all about the antibodies. That if the monkeys had neutralizing antibodies, they did well. But if they had a really good T cell response, they still got a lot of pox. They didn't die, but they really got severe illness. The caveat to the study is your classic animal model protective immunity study, which is you almost always go with a really high dose challenge because the monkeys are really expensive. So you really want to make sure all of them get severe disease. So I think the answer is if you really got a massive dose, right, of smallpox, the only way you were going to stay healthy is if neutralizing antibodies stopped it at the front door. But if you had a more normal, right, somebody coughed on you, it's a respiratory pathogen frequently, right? So if somebody coughed on you and you got and you got a little infection in your lung, and normally, right, when you get that disease, you don't get sick that quick. You get sick 10 to 14 days later or something. That's a pretty good amount of time for those T cells to actually respond and do something helpful, which wasn't really being tested so well in that model system, which are aspects of protective immunity that come up for coronavirus biology as well. So
this is a general problem with non-human primates. You have to use a lot of virus because, as you say, you don't have a lot of animals. We came across that in some of the SARS-CoV-2 experiments already. In fact, one set of experiments, they challenged by multiple routes just to make sure you get them infected, oral, mucosal, and intramuscularly, intrarectal, and so forth. So, yeah, it's what you're finding out is not always is going to be exactly the same as in people. Sean, I wanted to ask you, I wanted to remind or tell our listeners that you've written a book. You've written a biography of David Baltimore called Ahead of the Curve. You did that while you were with Raul Andino, right?
I mostly did it in college, and I finished it up in Raul's lab.
Yeah, it's a wonderful book, a story of his life. If you're interested in it, it's really good, and actually have a chapter devoted to my work, which is a chapter with very few words. It's called Interlude, and it's the sequence of the poliovirus RNA genome. which I thought was really funny. I remember Peter Palese showing it to me. He goes, "Look, you're famous now." In a book. Did you like doing that? Was that a lot of fun?
No, I wouldn't call it fun. It was definitely a labor of love. I mean, if you think about writing a scientific paper, it's like that, but 40 times longer, basically, and it's hard to wrap your head around. You can't just do it in the evening, sort of thing. You know, it really has to take a lot of your time. I'm certainly proud of it, and definitely lots of people have told me that they like it. I still have people write me and you know, want to get an autograph on it or whatever, which is wonderful. I wrote it really to write science for non-scientists, and use Baltimore's life as a prism to be able to talk about a bunch of interesting and important science. You know, so he's by talking about a single individual, it's possible to talk about molecular biology and cancer and genetic engineering and whatnot. So that was what was fun about it. And he was generous enough to give me access to his full archives at MIT. So it was really, as a historical project, it was great because there was all of this old interview material, you know, because as you guys all know, your recollections of events change as time goes on. So having a record of an event that's much closer, you know, to when it actually happened is much more valuable. Anyways, so that was the project. It took quite a few years.
So writing was actually formally part of your college degree. Is that right? Training?
Yes.
Nice. It's a great book. I recommend it to listeners. Ahead of the Curve. Really nice biography. A lot of the insight behind David Baltimore, who, of course.
So were you always
lots of fun virology in it? Lots of fun virology in it.
But I recommend you read the chapter called Interlude in very great detail.
Very, very careful.
So were you? When did you figure out that you were a scientist, and were you sort of struggling with, am I a scientist or am I a writer in college, or was that even an issue?
No, it wasn't. I went to MIT to do biology and research. I specifically wanted to go to MIT because it was one of just a couple of places in the country where it was very clear that undergrads could again get into real research. And so the writing came afterwards because of a recognition that, for me personally, a recognition that lots of non-scientists in general have to make all these decisions about science that they don't have the opportunity to understand, right? So they're being asked to vote on genetically engineered foods or just recombinant DNA technology in general, or cloning animals and whatnot. And so what are the things to help communicate that? I always tell people I got tricked into liking science. There was an NSF young scholar. I went to high school in the California desert, far outside of Los Angeles, and there was an NSF Young Scholars program centered in Los Angeles. And so I was still officially in LA County, even though I was like 2 hours outside of LA. And I got essentially bullied by my high school chemistry teacher into applying to that program. And those NSF Young Scholar programs are incredible, and I wish they had more of them for high school students, because the whole mantra of the thing is to take a high school student and pair them with a researcher somewhere at a college nearby, and just have them be in a lab and do something, and get together every couple months to go visit a college or hear a scientist or whatever, and at the end of the year present something. And there weren't high expectations, but it was about exposure. As you guys all know, science you learn in a textbook really has nothing to do with actually doing experimental science, and loving one, isn't loving the other. The only way to really know if you're interested in being a scientist is actually like try it out. And for biology, that's kind of hard to come across. So in my case, there were no colleges anywhere near me. So I think they accepted me to the program, and then they basically went, oh, wait, what do we do with this kid? There's no, so they assigned me to the furthest possible place from my house, which is Catalina Island off the coast of Los Angeles. And so every Friday I would get out of school and drive hours all the way across Los Angeles to the port, and then catch a boat across to the UCLA USC Marine Biology Lab on Catalina Island, where they have this protected cove and all this kelp forest and sea lions and stuff. And frankly, mostly I hung around, and I was like, wow, marine biology, this is science. Sign me up. I got tricked.
Nice. Cool. All right, Shane, let's talk about SARS-CoV-2. I want to kind of broadly talk about B and T cell responses, if you will. And I know you've done a lot of work on T cell responses, but I wanted to first get your view of the antibody response to SARS-CoV-2 infection. There have been a bunch of studies, and in my view, a lot of them are confusing. They seem to disagree. What is your take? What's the antibody response? And I guess, a mildly infected patient and a seriously infected patient.
So I think the short answer is it's great that there have been studies of, essentially thousands of patients at this point, right, on quite a few continents. I think people pretty consistently seroconvert fairly rapidly against the virus. And I'd say almost everybody who gets infected makes an antibody response against spike and a neutralizing antibody against spike, which is almost all of the neutralizing antibodies are against RBD, and that people with severe infections do have higher neutralizing antibody titers than people who have mild or moderate antibody responses. What are the things that you think are more of the conundrums there?
Well, I thought for a while some people were saying that a good fraction of patients didn't make antibodies, which seemed counterintuitive. But that it sounds like that's no longer the feeling in the field, right?
Yeah, so so you're right, Vincent, that was out there, and it does still get debated some, but I would say if you talk to labs who look at lots of patients and who are basically experts in serology, in the sense that they have sensitive assays set up, it's almost everybody seroconverts. Now, it's definitely a reality that a significant fraction of people have what I think everybody would agree is low antibody titers. And so if you don't run particularly sensitive assays, you can have people show up as negative. And that happened with MERS for sure, as well, that low responses were a thing. And so then it becomes, all right, do the low responses matter or not? And to me, the answer is that they, as far as I can tell, they matter a lot, because to me, the key is how much antibody do you need for protective immunity, right? And if the answer is it only matters if you have a huge antibody response, then fine. Sensitivity at the low end isn't a big deal. But to me, the monkey protection experiments, indicate that if you've got a neutralizing antibody titer in sort of the 1 to 20 range, that's still generally protective from disease, and therefore low antibody titers are relevant, which can be different for other diseases, right? Like I said, where low antibody titers can may be totally irrelevant. So, only being able to detect high amounts matter. But for this, I think it does matter to be able to pay attention to those low titers. Right. And most people become positive. There are some people who don't become positive, but that's anybody who tells you something always happens in immunology is either lying or wrong. And that's just not the way human immunology works. It's diverse by design, essentially.
So is there any correlation between the how good the antibody response is and how good the memory is, or do we even quantify that?
So it's a great question, and it's I'd say it's essentially ongoing. So in the general scheme of things, there is no clear-cut early marker for memory. either the B cell, helper T cell, killer T cell, or antibody side in humans. We would love for that, right? To be able to say, all right, 14 days into a disease, we can tell what a person's going to look like a year out. The rule of thumb is that the bigger the insult or the bigger the injury, the more likely it is your immune system is going to remember it, because it's just like brain memory. It's if you were at an intersection where you almost got hit and killed by a truck, you're gonna remember that intersection for a long time. You're gonna be careful about it, right? Whereas some minor thing, you just don't bother to remember. And to a decent degree, the immune system behaves that way. If the initial infection was really trivial, it's less likely the immune system's going to put significant energy into memory, because the memory does cost energy, right? But if it was a serious disease, usually the immune system's going to remember that significantly. But this is always an interplay between the immune system and the pathogen, right? And so does the pathogen try and short circuit that in some way, right? And so those are things you have to learn. So I guess then the answer becomes, we just have to wait. And so right now, waiting means people have data about 90 days out from people. More than that serologically. They have like 120 days out serologically. But on the cellular side, Marion Pepper put a preprint up that's the best data to date for looking at a first look at memory. And it's basically 90 days out, which is not long enough to really tell what's going on, but it's a really helpful step along the way. And it's very good to see that they were seeing memory B cells, memory helper T cells, and memory killer T cells in most people at that 90-day. Obviously, if it was already gone at 90 days, you're not going to be seeing it at 6 months. But I would say 6 months would be the benchmark we'd really like to see.
Yeah, it's a common feature of the common cold coronas that memory wanes, I guess, within a year or so. Are we expecting a similar thing to happen with SARS-CoV-2?
So I'm a little bit of an outlier in that argument. So I disagree with that statement, actually, fundamentally. And to me, it pretty much all ties back to the one clear paper on this, right, which is the English Common Cold Unit study, where they infected people with a common cold coronavirus, and they got infected and sick, and then they had them come back a year later, and they re-challenged them. And the abstract of that paper says, they weren't protected. They got reinfected. And you take a look at the data, and the data is 0 people got sick. Nobody got a cold again. And so, yes, they had at least something that they could PCR amplify from the back of somebody's throat, 24 hours after the challenge. So it was not perfect sterilizing immunity, but none of those people got sick. So I think there's a lot of uncertainty there, because common colds haven't actually been studied that much over the decades. There is definitely a decline in antibody titers between like a re-challenge like that and a year later, but all those people are still positive for antibodies. I mean, like, if you go out right now and do a serosurvey of the place you live, right, you would find that over 90% of people are positive for antibodies to at least 3 of the 4, if not all 4 of those common cold coronaviruses. And in fact, I mean, data I've seen indicate that people stay pretty stable at those levels for long periods of time. And so there, then I think, to me, you get into more sophisticated arguments about, right, protective immunity from disease versus protective immunity from any detectable virus, and what do the two mean for what are you trying to discuss, right? Protection from disease or any detectable virus, and does that affect transmission or not, which are more complicated. I'm actually of the opinion that it's not that immunity just disappears in a year from common. I'm not convinced by the available data.
Yeah, I mean, many people in their papers say that, right? And maybe it's an assumption that's made, and that shouldn't be. You could be right. I just don't know.
How about
it's essentially all comes from that one paper.
Really?
Oh,
How
yeah.
about,
It's all literally from that one paper. And that one paper shows none of those people got colds a year later.
Yeah.
how about antibody titers to SARS-CoV-2? Are they pretty stable? Do they decline? And what's the significance?
So that's a debate right now. There are preprints and papers on both sides of it. My assessment so far of the totality of the data are that the kinetics of the antibody response look pretty normal in your average COVID-19 case. That is that people make a response, and if you look 90 days after the peak of the response, 90 days after the acute infection, the titers are probably somewhere between stable and like a fourfold decline from that peak, which is all very much in the normal range of what you see to most viral infections. There's a range of outcomes, and so it's unclear if from different studies, and so it's unclear if some patients are significantly different from other. But to me, the totality of the data largely look like that. It looks mostly normal to me. And again, to me, what matters is how much do you really need for protective immunity, right? And if the answer at the end of the day is you need a lot, then a couple-fold decline becomes a really big deal. But if it's actually you only need a new titer, something like 1 to 20 to prevent disease, not necessarily infection, then those declines aren't as worrisome. I think the clearest data on poor responses or things at the low end are this Nature Medicine paper from China, where they had a pretty clearly defined group of asymptomatic people, which in their study was a pretty small fraction, right, of the total people who got tested. It was maybe 15% of people fulfilled that criteria. And when they compared those people to other people, they said, okay, those people in general became seropositive, but whatever it was, something like 60, 90 days later, I think 40% of them were negative for detectable antibody at that point. And so the conclusions that have been made from that are that asymptomatic people lose antibodies faster. And I don't think the data wasn't showing that, in my opinion. The data was showing they started lower, so they became negative faster because they didn't have much to start with. But that's of the subgroup of people who were asymptomatic. And so, yeah, maybe it was just such a small exposure and response. It just didn't trigger much of a fight by the immune system.
So you mentioned that, 90 days is sort of as far out as we know right now. And, my impression would be that we need to look a little longer to sort of fully understand some of this. I was really interested in the germinal center paper that just came out showing the lack of germinal centers. And so how long do you think we should look?
I think really the vaccine literature is a good place to look for that. So when people do like their clinical trials of vaccines, and they say, see, we got an antibody response at 30 days. That's basically not predictive of what they're going to see at 6 months or a year. But in general, what you see at 6 months is predictive of what you're going to see at a year and longer. And essentially, a decent amount of that is just built into the nature of the kinetics of antibody responses. You need at least 3 time points, okay? Because when you initially make a whole lot of antibody, if you then stop making a bunch of that, those antibodies are going to decline with a half-life of about 21 days, right? So you have half as much 3 weeks later, and then half again as much 3 weeks after that. And so you have to kind of let that go away to then be able to see what's the steady state amount of production that your body is making that's going to remain. So if you just have 2 time points from peak and then some late time, you're going to see you might see some really extreme slope, right? But you need an intermediate time point to see, oh, actually, it was coming down pretty fast and then it was flattening or not. And so I think the best data will be when people have 6-month time points and you have 3 time points, some early time points, some intermediate time point, and that's 6 months to see, are you just constantly losing the response or do you really have something that looks like it's going to be some memory up to a year or longer? And for classic SARS, it looked like there were responses out, say 5 years or so.
Go ahead, Rich.
Do people make antibodies to viral proteins other than spike? And are those responses important?
They certainly make responses to other viral proteins. I mean, my rule of thumb for talking to people about that is that, generally speaking, the immune system is blind to which parts of the virus are going to be neutralizing antibody targets. It can't tell, right? All it can tell is that there are these foreign proteins. And so essentially then the immune response needs to try and make antibody responses to as many different proteins of the virus as possible to give the best chance of getting a neutralizing antibody response. And frequently those responses correlate to the magnitude of the viral protein that was around. And so it's really common that things like nucleocapsid, right, or core proteins are really strong antibody responses, whether we're talking about a coronavirus or HIV gag or whatnot, which is why those are frequently quite good serodiagnostic antibodies, is because they're frequently high level responses. They're not generally considered to be relevant for protective immunity. The bar is higher for showing that. I mean, you basically need to show that you can take a monoclonal antibody against a non-neutralizing target and inject it in a mouse or a monkey and show that it has some positive benefit. Otherwise, really neutralizing antibodies are generally at the benchmark. If you can show neutralization in vitro, it's probably helpful in vivo.
So let me ask you, and again, I might be wrong on this, so correct me. So I've read some of Stan Perlman's work on SARS, the original SARS. He says, if you look 15 years after, these people have no memory B cells, but they have plenty of memory T cells. So do you think that's correct? And if so, what are the implications for SARS-CoV-2?
That's correct, Vincent. And definitely Stan Perlman has a very good sense of the literature, of course, as being one of the main people who's really been studying coronavirus immunology all along. There were 2 different studies that looked for memory B cells greater than 10 years out, 2 classic SARS, and neither of them really saw anything. The exception to that is there have been people who've tried to see if they could find neutralizing antibodies in a classic SARS case, right? That would be a cross-reactive neutralizing antibody. And if they look deep enough and hard enough in those people, they could pull out a few cells. But those are pretty, intensive searches. And I don't recall seeing any evidence of circulating antibodies in those. So that's correct.
And can we make any predictions about SARS-CoV-2 based on that, or we just have to see?
Yeah, we have to wait and see.
Okay. It's fair. Sorry to put you on the spot, but
no, it's a legit question. But I would say while they are related viruses, they are different enough such that I wouldn't. Like if you told me what the memory B cells were for one of the common cold coronaviruses and asked if I thought for one of the other common cold coronaviruses, would it look similar? I'd say yeah. But since the pathogenesis of classic SARS was so lethal and spread so differently, I think the memory may in fact be.
Yeah. Okay.
I wouldn't make the assumption that the memory will be the same.
I'm afraid we have to move on to T cells because we only have 15 minutes. I'm sorry. And I know you've done a lot of work on T cells. So why don't you have two main papers. Can you summarize them for us, and then we'll chat about them a bit?
Yeah, sure. So really going back to March, there were lots of uncertainties about and lots of fear people had about just were people making immune responses at all to this virus, or was it really strange? And then obviously there was a huge amount of effort to try and get vaccine development going as quickly as possible. And Alex Setti here at LJ and I both realized that it would be very valuable to have a benchmark study of what's an immune response look like in average cases of COVID-19, both for understanding the clinical disease and for understanding what was the likelihood of being able to develop a vaccine. And so for us, that comes down to measuring all three arms of the adaptive immune response, right? And to me, it's really key to measure all three at the same time, the antibodies, the helper T cells, and the killer T cells. And we felt it was key to do that in average cases of COVID-19 first, which is to say people who had something like flu-like symptoms and recovered pretty quickly without having hospitalization. First of all, because those are average cases, so it really makes sense that should be your starting point as a benchmark for understanding the immune response to this virus. But also, those cases probably reflect development of protective immunity or a good immune response, right? Because the virus infected, got ahead of them, they were sick, but the fact that they weren't sick for very long and they got better probably meant that the immune response caught up to the virus and cleared the virus, and that's a good immune response. Now, we're having to make some assumptions there, right? Assumptions that we wouldn't make if it wasn't 2020, but they were reasonable assumptions given the time. And we wanted to look for T cell responses across the entire virus, which is a hard thing to do, but Alex Setti's really probably the number one person in the world at predicting T cell epitopes. And so he had already gotten going on that. And so we felt like we could do this rigorously, because part of the problem with T cell responses is that it's pretty easy to miss them, that you could detect them in one person and not another because you're looking at the wrong thing. So we did that. And basically the bottom line was we took 20 convalescent cases, right? So people who had recovered from the disease, something like 30 days out of average cases, and just said, what's it look like? And basically we saw, we considered it good news that they all made IgG responses to spike. They all made helper T cell responses, and most of them made detectable killer T cell responses. And the helper T cell responses were also against spike. And the T cell responses were well distributed across the genome, and the T cell responses look like functionally they did the things we would expect a good antiviral immune response to do. And the antibody responses and the T cell responses were of a reasonable magnitude, basically. So like all those things pretty much across the board, we considered good news and good signs that this was largely what we would expect a person. If you just told me there was a new respiratory virus, right, and just told me no more information than that, what type of immune response would I expect if it was a good immune response? It's basically what we saw. And so that has helped inform studies of in people who got very bad disease, right? People are hospitalized. Is there something strange that goes on in immune responses there? And we can talk about that. And it's also informed a lot of the vaccine studies. I mean, that the paper has been very widely cited in the vaccine field for saying, okay, it looks like an antibody is important, antibody response is important, but it looks like it's also normal to elicit a T cell response. So do you get a decent T cell response to vaccine X or Y? And our biggest concern going into it would be that there would be a terrible T cell response to spike, and all of the vaccines were really focused on spike. And so that would be a huge red flag. But what we saw was that there really were reasonable T cell responses to spike. So while I don't think a single protein vaccine is necessarily perfect, it seems like an entirely reasonable approach in 2020.
So I guess you would expect CD4 T cell responses because you're getting antibodies, right? You need those. But the CD8s, is anyone looking at their function? Because as you know, they have to kill infected cells, and we don't know if they're doing that or not.
Correct. And so what you normally do is you look at surrogates. So do they make granzymes, which are the pore forming proteins that'll kill the target cell, or do they make interferon gamma, which is just a good general surrogate for them being functional at all? And they've generally looked fine by those metrics. Yeah. Yeah, good question, Vincent. Yeah,
I was raised in the era where Rafi and others would fill cells with chromium and look at CD8s, pop them open, which people don't seem to do anymore.
It's a really messy assay.
Well, there are other ways to do the same thing, right? But the point is that I understand the surrogates, but I'd love to see killing of virus-infected cells at some point, right? Or maybe you don't think that's necessary. I don't know.
Yeah, it'd be nice to see. It's in general, the molecular markers do correlate very well. Okay. And that what you see is you can run into cases where you don't have those molecular markers, and then definitely killing function is not okay. But if a CD8 expresses perforin and granzyme.
Okay. Now, in your first paper, you made the suggestion that there might be some cross-reactive T cell epitopes with common cold coronaviruses. And then in a subsequent paper, you extended that to actually showing that there are memory T cells. These are in people who have never seen SARS-CoV-2, yet they seem to have memory T cells that recognize T cell peptides. So what does that mean?
Yeah, so that was definitely the biggest surprise that we came across was when we looked at our control donors, our healthy control people, we could see some measurable T cells there, and that was hard to decipher or be convinced of initially, and so we definitely ran the experiments a bunch of times, and luckily the way we had set it up was that we were using healthy control donors from basically 2015, and so these are people who we knew for sure there was no way they'd ever seen SARS-2, and yet we could detect T cell responses from them. And what was interesting was that indicated that those people already had memory T cells that could recognize SARS-2, even though serologically, right, people really don't have cross-reactive neutralizing antibodies, and most people really don't have antibodies that can, for example, see SARS-2 spike protein. But here we were saying about 50% of people seem to have memory T cells that could recognize SARS-2 epitopes. And the reason that was interesting and we wanted to get that out there as quickly as possible was really, it was really two things. One is that just based on general immunological principles and things we know about flu, if you already have memory T cells at the time you get infected, those memory T cells, those helper T cells, should help you get a better, faster, stronger antibody response than you would if you didn't have those memory T cells, as well as those memory T cells may directly have antiviral function. And so we speculated that could mean that if you have those memory cells, you would have less severe disease than somebody who didn't have those memory T cells. You would still get infected, but instead of your immune response taking, let's say 7 days to get up to speed and start controlling the virus, maybe you're controlling the virus after 3-4 days and you never even noticed that you got sick, right? So instead of having flu-like symptoms, you have an asymptomatic disease, or instead of having a fatal infection, you may still have a hospitalized infection, but you still did better. You don't die. That might be what we would speculate having memory T cells of that type would do for you. And so then the follow-up paper in Science, which was very much led by by Alex Setti and junior faculty member Daniela Weisskopf here at LJI, showed that yes, these are indeed direct confirmation that they were memory T cells, and direct confirmation that at least a lot of them are from common cold coronavirus infections. So as we were talking about, there are 4 common cold coronaviruses that circulate widely. And it looks like what happens is that something like 50% of people happen to make T cell responses to those common cold coronaviruses that can cross-react against SARS-2, and the other 50% of people don't, or it's a timing difference. That has been reproduced on multiple continents now, so that's not something special about people in San Diego. And so there definitely are memory T cells. They definitely are cross-reactive from exposures to common colds.
Am I correct?
But what we don't know is that they have any functional relevance. So there's no direct evidence that we have or anybody else has that these memory T cells are actually doing something. So again, just based on general immunological principles and some things we know about flu, which I can give you specifics of, we would hope or speculate that they would be a positive, but it is entirely possible that they have no impact on the disease at all, or that they have a negative impact on the disease, that they could be screwy in some way because the immune system is complicated. I will quickly mention the second thing, the reason we put it out quickly, and then I'm happy to open it up to discuss because it's really a fascinating topic. The second reason we wanted to put it out quickly is that in phase 1 clinical trials for vaccines, you're frequently looking at like 20 people, okay, and asking, did your vaccine work or not? And clearly there were a lot of phase 1 clinical trials going on in different parts of the country with different vaccines. And we said, well, wait a minute. If these memory T cells actually are functional, you could just happen to get 20 people who have those memory cells or don't, or predominantly do, and you get a good response to one vaccine, you get a lousy response to another vaccine, actually just because of the composition of the people you looked at. And so we started telling vaccine people very quickly, look, you really want to look at those baseline responses, like check the people before you immunize them and see if they have any T cells. Because the expectation was nobody needed to look at them because nobody's ever had SARS-2 before. So of course, any response you would see would be de novo. And we said, well, it looks like it might be more complicated than that. And so those studies are going on as well. Okay.
So they listened to you. They basically listened to you, right?
Yeah. As far as we can tell, they're doing those studies.
Okay.
Yeah. It seems like in one of the papers you said that was important for phase 1 trials, but wouldn't it also be important for phase 2 and phase 3 when you're getting more efficacy data over time? I mean, it seems like with more numbers, you might have better data.
Right. So it's a really good question, Kathy. At the end of the day, the phase 3 trials, the endpoint is do you get protection or not, right? And so it needs to protect overall in the population, whether or not the people had pre-existing memory cells. Scientifically, it'll be totally interesting to study that in the larger clinical trials and understand, can it explain, are there differences between people that can be explained by whether they had cross-reactive memory to common cold or not. The big concern from us was in the phase 1 trials, when people would making decisions about which vaccine candidates to go forward, you could be misled by simply the small number of people involved and some randomness to who might have some memory.
Right. So I have 2 questions. In your paper about cross-reactive T cells, you define cross-reactivity as sort of 67% in terms of similarity. And I wondered what was the rationale for that 67%?
Right, really good question. So essentially, there wasn't a rationale. That was basically a guess, an observation, and then a feedback loop. And I think Alex, we tried to be clear about that in the paper, but obviously those papers are really short, and so we probably weren't. T cells, how much similarity would a T cell, would an epitope have to be cross-reactive between 2 different viruses? And the answer has been we don't know. The study lab did a nice flavivirus study where you guys know there's been a lot of interest in different dengue viruses, and then Zika, which is related to the dengue viruses, and well, how much relationship matters. And so on the T cell side, they were trying to get at that. And they saw that something in that 67%, that 2/3 range, seemed to be an indicator of an epitope that was more likely to be cross-reactive than not. And so in this study, we first made some pools that just had anything with homology. And then we stratified them into pools that had high homology or low homology. But really we did the experiments were done by Daniela to just look at everything. And then when looking at the T cell lines for which ones were cross-reactive, if they were lower than 67% homologous, almost none of them were cross-reactive. And of the cross-reactive ones, I think all but one had greater homology than 67%. And so then it could sort of feed back and say, okay, that looks like a reasonable benchmark that.
Got it. So then I guess bigger picture, it seems like, I think you mentioned before that pretty much everyone is infected with one of these common cold coronaviruses. And so how do you think we're seeing, differences in T cell responses, and how might that influence what we're seeing with heterogeneity in COVID?
Right. So this is one of those. This is one of those situations in science where you have an observation that could basically explain everything, or it could literally explain nothing. And we honestly do not know. We do not have the data. The it could explain everything scenario that lots of people have been interested in speculating on. And again, it's fine to speculate on it. And the problem is some people have really declared it knowledge, and made some really dramatic claims, saying that this is known. That's a huge problem, and we can come back to that in a second. But it could explain everything scenario would be to say, oh yeah, well, it's been interesting that COVID-19 has had such a broad spectrum in terms of disease severity, right? Why are there so many people who get very mild or asymptomatic, and some other people get really severe infections? You go, well, okay, maybe it's people who have some cross-reactive memory that give them just a little bit of a head start in that immune response, and that's enough to just blunt the infection, not stop everything. Or that, why is it different in kids? And somebody like, well, of course, kids are filthy. They're getting common colds all the time. So do they just have more of these responses? So, I mean, those speculations can go sort of ad infinitum, right? And some people speculate like, oh, in Southeast Asia, maybe there are more coronavirus infections that are different or related. And so maybe you actually see milder diseases because of that. Again, no data to support any of that, but those are each questions worth exploring, right? And you've got to start somewhere. The biggest problem has come from, there have been prominent public figures who have misunderstood or mistaken the immunology to say, oh, these guys saw 50% immunity in people. And so therefore 50% of people are already immune. So if you get hurt, if you need 70% to get herd immunity, the 50% are already taken care of. So you just got to get to 20%, which is New York City, right? And then you've totally hit herd immunity. And that's just a fundamental misunderstanding of the data. There is no way those 50% of people who have memory T cells are protected from getting infected. In general, what T cells do, right, is once you are infected, they can help control the infection quicker or blunt it. And we even said, one very good scenario would be that those T cells would blunt the infection, but they would do it so slow that it really wouldn't affect the viral loads at all. Right? It may keep you from feeling bad, but it really might not, in terms from an epidemiological standpoint, it might not change people's viral loads at all. And again, it might help explain stuff, but it wouldn't change it. Anyway, I'll pause there.
I'm glad you.
And I will have to go in a few minutes.
It's okay.
I want to ask two basic questions, because as a not so much of an immunologist, I have wondered about two aspects of this, and having an expert now on it. How are these epitope pools designed? If you could just give us a simple explanation of the algorithms for how you predict those epitopes, that would be great.
Sure. So this is absolutely what Alex said he has devoted like his entire life to, and he's amazing at it. And so, like I said, he's the world expert in this, and that's what made it possible for me to help out on these studies. Was he was already so far ahead with predicting epitopes, right, to SARS-2 that these experiments were possible. The way it works is so T cells are HLA restricted, and each person expresses potentially like 6 different HLA molecules, right? So these are the major histocompatibility complex genes. This is, right, simply put, right, this is why you and I can't swap kidneys, right? It's not the kidneys are totally functional, but your immune system sees them as different because they express these different molecules. And so there are lots of different molecules of those, and those are the things that present the epitopes. And so different people will present different epitopes, and those epitopes have different sequence motifs that will bind to different HLA molecules. So what people like Alex said they do is they say, okay, actually a bunch of these HLA molecules are similar. They bind similar things. And if you take everybody in the world, how many different HLA molecules do you have to look at to represent things that would cover like 98% of people, okay, to simplify it down from having to look at the hundreds of different HLAs. And so they do that. And then frankly, it's a data-driven algorithm. You just try lots and lots of different peptide sequences, and do they bind and with what affinity? And then you just line up all those sequences, and you ask a computer algorithm to say, okay, given this baseline data set, what's the likelihood that this new peptide would fit that? So based for humans, there's a huge data set.
Okay, so it's based on some empirical data of things that are already known, and then refined from that. Okay, that helps a lot. Okay, and then
thousands of them.
Yeah,
maybe tens of thousands at this point.
Okay, and then secondly, if you could describe the T cell assay that you do. I mean, so Vincent alluded to the older types of functional assays, but the ones that you actually did in this paper, or these papers.
Yeah, that's a really good question. And that was actually a key decision we made. So I'd say that the classic T cell assay is really a cytokine assay. And really it's the intracellular cytokine assays that have really been the benchmark, where T cells, when they see their target, they will make cytokines. And then if you put a plug in the T cells, they make the cytokines, but they can't secrete them. Their job normally is to secrete the cytokines, but you basically put a plug in them and they can't secrete it. So then they build up the cytokines, and then you identify the T cells that physically have cytokines inside them. And those are the ones that are virus specific. Okay. Wonderful assay, been used for 20 years. The problem is with the helper T cells, they frequently don't make your favorite cytokine. And they frequently don't make your favorite cytokine very quickly. And so when you look for cytokines, generally you have to bring some of your own expectations to the assay. And on the killer T cell side, it's not such a big problem because they'll basically all make interferon gamma. But on the helper T cell side, it becomes a problem when you miss a lot of things. And so instead, we used what we call a cytokine agnostic assay. We tend to call them AIM assays, for activation-induced marker assays. So the T cells will always do something when they see the viral antigen, but they don't necessarily make cytokine or your favorite cytokine. So could we capture a broader pool? And so that's what we look for, basically surface markers that the cells will upregulate. And a lot of different labs use these assays now over the past few years, and they're quite helpful to look in a more agnostic way. They're also a live cell assay, to just get to a technical aspect of it. If you do an intracellular cytokine assay, you've defined what that cell is, but now you can't do anything else with it because you had to kill it to plug it. Okay. But these instead are live cell assays, so you can actually sort the live cells and ask, okay, what's the whole gene expression profile, right, on that cell, which has a huge value. So that was the nature of the approach that we took. And we find it to be more sensitive than cytokine assays, but T cell, your personal experience may vary. T cell assays definitely vary from lab to lab because they're live cell assays. There's
And then it seemed like you then did assay some cytokine production, at least for some.
Yeah, we did, because we definitely want to know those functionalities, and we did it two ways. We felt it was important to compare those sort of AIM assays versus conventional intracellular cytokine assays, and basically our experience was the AIM assays were more sensitive than the cytokine assays, but told the same thing. And then we did more general cytokine assays, so that the T cells, if you don't mess with them, they'll just secrete the cytokines into the culture supernatant, and you can measure them there. And so we did that. And the cytokines involved largely looked like antiviral cytokines. Now we're doing all of that again. We've been doing all that again with hospitalized patients to try and understand, because there's been a lot of debate about immune responses in hospitalized cases. Are they good or bad? And to make that understanding, you really have to be able to distinguish innate immune responses from adaptive immune responses. And for us to be able to measure, to make conclusions about adaptive immune responses, you really need to measure the antigen-specific T cells. And there's not much data out there currently with hospitalized patients that have measured neutralizing antibodies, antigen-specific helper T cells, and antigen-specific actual virus-specific helper T cells and killer T cells. There have been a number of studies that have used surrogates or sort of bulk measurements, and that can get confusing because people would just say, "Oh yeah, we looked at the T cells and they were doing this." And you're like, "Well, did you look at the ones that are actually SARS-specific?" And the answer is, frequently been no so far. So we're trying to help fill that gap.
Shane Crotty, La Jolla Institute for Immunology. Thanks so much for joining us. Appreciate it.
Thanks. It was a real pleasure. Excellent questions across the board. Take care. You guys are doing great stuff and a really valuable service.
Thank you. Bye-bye.
Thanks, Shane.
Thanks, Shane.
Thanks. It was great to talk with you.
Bye for today.
All right, let's summarize that.
Another winner.
Yes.
No, that's not what I meant, but yeah. So every SARS-CoV-2 infected patient seems to make some kind of antibody. Yep. We don't know how long they last because it's too soon. Our assumptions about common cold coronaviruses may not be correct. Is that all fair?
In that, go ahead.
Responses go away.
Right.
Yeah.
But that maybe nobody gets sick.
I don't know. The sick part, I don't know, but the antibodies may not go away completely. I mean, so many papers say they go away within a year, and then you get reinfected. I think Ralph said that the first TWiV he was on this year, and then many other people said it. So maybe that's not correct. I don't know.
And according to Shane, that's all based on this one study in England that gave evidence that individuals could get reinfected, but hidden there with none of them were getting sick. And this strikes me as people don't readily distinguish between whether you're getting infected and whether you're getting sick. Okay. And this shows up, I think, most damagingly in this idea that since kids don't get sick, you can send them all back to school and everything's okay.
That's right, because they must not be getting infected, right?
Yeah, right. Which is nonsense.
Yeah, I feel like that's one place where immunologists could do a lot to communicate better that issue.
All right. So then T cells, these mildly infected patients, which is all that he's looked at so far. Be interesting to see his other study with the seriously ill patients. Mildly infected patients, they make a range of CD4 and CD8s against viral protein, but peptides in viral proteins. And the interesting observation is that some of those seem to cross-react with common cold coronas, but what that means is anybody's guess, as he said, right? And I think people are going to want to find out if that means anything, and the experiments need to be done.
And I think the other thing that he mentioned that's really important is that we know that those cells are present, but we don't know necessarily that those cells are actually having any kind of function in protection or anything like that. So the function part of this is not really well understood.
So the funny thing is, and he mentioned this, there was one doctor on Twitter who said this, oh, we got 50% immunity already. And I'm like, what are you talking about? I'm often astounded when people completely misinterpret things, but I guess I shouldn't be anymore, right?
Right.
Oh.
Right.
Another take-home message that I got from Shane is that the I don't know that it's the major, maybe it is the immunodominant antibody response is to spike. Okay. Mm-hmm. And certainly that's the major neutralizing response. And that may be sufficient.
Well, that's driving the vaccine development, right? That's
exactly.
why it's all the spikes.
Yeah. Right. He didn't see, I didn't hear him saying, these spike only vaccines are a disaster. Okay. He seemed to think that was not an unreasonable way to approach.
But you know what? The spike decision was made before we knew that the immune response was spike focused, right?
Right.
I'd rather be lucky than good.
Right? Fine. It's fine.
One thing that I liked is that I've always divided the adaptive immune response into two, and he consistently divided it into three. That was interesting to see. So the antibody response, the helper T cell response, and the killer T cell response.
I'm feeling a little bad about sucking up so much time on vaccinia, but that's
it's okay.
Actually interesting because I think an important lesson that I keep having to relearn is that vaccination and vaccinology up until very recently was entirely an empirical discipline. Mm-hmm. And when he started working on vaccinia, I meant to ask him whether, I'll bet you some of that was biodefense stimulated, right? Because pox viruses for 10 years or so after 9/11 got a biodefense bump. We'd eradicated the disease and we didn't know why. Okay, we didn't know what the immune response was or what its relevance was, and it's interesting that he finds that it's basically an aggregation of different antibody responses in vaccinia, but I wouldn't necessarily extrapolate that to another virus because vaccinia is complicated. Okay, and has many different ways of attaching to cells and being taken up rather than just this one thing that attaches and gets sucked up. So it's not, I don't think you can extrapolate from the experience with vaccinia to necessarily to another virus, but it's interesting.
That also has about a gazillion genes, right?
Yeah.
And a gazillion targets.
Yeah. So that what he talked about there is interesting because he said, if you give non-human primates a lot of virus, you need to have antibody, but it could be that, in a normal infection, T cells might be enough to limit infection, right? Because it would make sense that if you got a few cells infected, the CTLs go and kill them, boom, that's the end. But if you're putting so much virus in, there's no way that can happen. So really another caveat for animal models, right? When we have to give them so much virus because they're precious and I like that a lot. I think that was so. In that sense, Rich, no question is really on the side. It's all intertwined. I like it all. It's all good.
I'm astonished that I can have these conversations about immunology.
Hard.
I've learned so much from this.
Yeah, but Rich, when they leave, they laugh at us. you and me anyway. What are these guys' questions? Anyway, I'd like to hear the result of the serious infected patients. It's quite interesting. The thing is that in an evolving pandemic, the first few months, it's a mess because every paper, and there are too many papers coming out, they're slightly different and people get confused and say, oh, it can't be. And now it's starting to sort out. So that's good.
And you know what? It's only been 6 months. No, wait a minute. It's been longer than that. Where are we?
7.
End of August?
Yeah.
8 months.
7 or 8.
Yeah. Okay.
One thing that's a little bit different to me is that it seemed with Zika that early on there were way more review articles than there were primary papers. At least with this, There's lots and lots of primary papers and, maybe a relevant number of review articles. But that kind of drove me crazy with Zika.
It's just every new outbreak that's crazy. Yeah, lots of stuff just pouring out, and not all of it good, unfortunately. I think the fraction of work that's not as rigorously done goes up because I think it's done quickly, and people think that it's important just to get done. But I think it's better to get it right.
Definitely.
That's okay. They have to. All these people have to go through the TWiV filter eventually. Okay, we're the peer review. Us and Twitter, right?
Right.
I don't know about Twitter. I think Twitter can be a big problem. Speaking of Twitter, so Amy and I were discussing the other day. As we talked about previously on TWiV, I think with Daniel, the convalescent serum trials didn't look good, and so FDA was considering issuing an EUA, and Fauci and Collins said, no, we don't think there's enough data. And then the next day, the FDA issues an EUA. Okay. And then a few days later, the head says, I made a mistake. And where does he announce this? He announces it on Twitter. Instead of doing a press conference, would be widely picked up, he announces it on Twitter, which I think, I agree, is just not the place to do that. If you're serious about something, do a press conference. Because if you do the original EUA in your normal fashion, you should reverse it too. But I think...
Well, he's taking a hint from how the president does it. Yeah.
Yes, but I don't think it's necessarily a good... You can have your conversations, but if you want to make policy, I don't think it's the place to do it.
Right.
But more broadly is that this intersection of politics and science, right? I mean, obviously, the administration has an agenda to push vaccines and antiviral therapies to make them look good, but that shouldn't drive it. It should be the science that's driving it, unfortunately. And I know that we have an intersection between society and science, and science has its own view, and society has an agenda, obviously. But I don't see why an administration can tell the FDA or anyone, NIH, CDC, what to do. It doesn't make any sense to me.
There needs to be a collaboration between society and science rather than a conflict.
Yeah. I was really sort of taken when Daniel talked last time about the issues with randomized control trials when different treatments were thought to be the best or terrible. He couldn't get people to enroll in the trial that he needed to have great data. And so when I saw some of these convalescent serum announcements, I thought back to that conversation because there is no randomized control trial data on convalescent serum. And I would assume this will make it much more challenging in a similar way.
So also the things that Daniel said about the dangers of that type of therapy, where the coagulation factors are a real problem.
No, that's a great point. Especially this is a disease where clotting is an issue. Right?
Right.
Yeah.
Maybe.
I mean, he made that point so clear.
Yeah.
Yes.
Good.
Coagulopathy.
Very good, Davidson.
You got to slow down and you can get it. All right, let's do some email. Claudia writes, hello, TWiV people. Regarding those never-ending ongoing stories like the Florida sheriff and so many others of people not wanting to follow common sense rules for political or whatever reasons, it reminds me of some long ago wartime stories. In World War II, when Germans started bombing London, when the alarm went off at night, people were supposed to shut down their lights. A total dark city would obviously make it more difficult for the enemy to spot their neighborhood and hence to bomb them. If you, for any stupid reason, would be the person to say or even think about something like, quote, I have the right to keep my lights on, unquote, well, you can imagine what would happen. So, like Vincent says, this is war, people.
Cheers. That's a really great analogy.
I like that very much. Cheers from Brazil. It's a great point. I like that very much.
I wonder if that's there's probably lots of Claudios in Brazil, but I know a couple of Claudios in Brazil.
I never know. The last one you got right.
Yeah.
That one guy you said.
Bradley from Gainesville, with whom I have now reconnected, as with a few others in TWiV, and it's been very nice.
Brianne, can you take the next one?
Sure. Charles writes, "Hello, TWiVers. Just a pissed off human living in Chapel Hill, where the weather is nice at 85 Fahrenheit, 29 Celsius, with a chance of rain later today. I did listen to all of 654, as I'm sure most of your listeners did. I just sent the following short and to the point correspondence to my Congress critters. I hope to get a more meaningful response this time around. My first response from Senator Tom Tillis talked about what a great job we have done. In other words, pure BS. Senator Burr was not much better. He talked about how much PPE we produced. Representative David Price was a bit better, but not good enough. Thanks, Charles. P.S. about the ignorant sheriff and chief of police. I have lived in some diverse places, Lexington and Murray, Kentucky, Burlingame and San Francisco, California, Durham and Chapel Hill, North Carolina, and Ocala, Marion County, Florida, just south of Gainesville. I would live in any of them again except for Ocala. The sheriff banning masks and the chief of police deciding which laws to enforce without being fired should tell you all you need to know about that area. PPS, because of the language in this email, Dr. Brandbarker may want to read this one. If that is a microaggression, sorry. PPPS, a question for Twig. Salt hay had me baffled. I assumed it was hay that had salt added to it to keep seeds from sprouting. Turns out, I think it is salt marsh hay, which needs salt to sprout. Is that correct? Do you mean Spartina?
So that's actually Dixon adding in, do you mean Spartina? And that was enough to put me on, send me off on the Spartina trail. So salt marsh grass is Spartina patens. It's also known as salt meadow cordgrass and also known as salt hay. It's found from Newfoundland all along the coast of the eastern United States to the Caribbean and northeast Mexico. And so as to his question about it needing salt to sprout, from at least some species that I saw, salt is not necessary for the sprouting. The plants are salt tolerant. And in fact, if they have too much salt, it can make them stubby.
So this is because I went on This Week in Gardening, Twig. We went and bought some salt hay to put on top of the bed so that it would suppress the weeds. But one year we bought hay from Home Depot, and it was full of seeds, and they all sprouted. So, it defeated the whole purpose. So we found out that there's this salt. I don't know how it is. All I know, it was $25 a bale, which seems to me a lot for a bale of hay. And I don't know if it's Spartina or salt-treated hay, but it's expensive.
Well, if it's Spartina, it sounds like the only issue with the salt is that it grows in a marsh. Mm-hmm. It's not as if it's got salt in it when you spread it out. Yeah. But maybe since it grows in a marsh, it's not going to bring into your garden the sorts of weed.
Maybe.
I'm totally making stuff up now.
Yeah, no, I think it's probably just a good solid mulch and doesn't have a bunch of seeds in it.
And it's expensive because they drug it into your New Jersey garden from a salt marsh.
Right.
Maybe. Well, it does work because it's still there and it pretty much suppresses the weeds and nothing grew in the hay itself. So that's good.
And Charles seems to have given us his letter to his Congress critters to go on our letter page.
Okay, we'll put that.
It has a couple. Yeah, and it has a couple of. He said short but sweet. He's got a couple of all caps things to make his points.
Okay.
Yeah.
Kathy, can you go next?
Sure. Esapeka writes, Dear TWiV, I have a fairly simple question that came to me while listening to TWiV 652 about transmission of SARS-CoV-2. Is it possible that the incubation time for the onset of symptoms and for transmissibility are decoupled in SARS-CoV-2? I suppose with my limited understanding of SARS-CoV-2 biology that there may be a component that suppresses the immune response, which causes a period of asymptomatic transmission. Could the level of immune suppression and therefore the delay from peak transmissibility to symptom onset be a significant indicator of disease severity? I'm a humble non-viral structural biologist, so I might be overlooking some obvious facts here. Regarding home testing, I hope the preprint below has caught your eye. In it, they describe an open source application to produce a LAMP-based test that can also be used at home. And this test is a test from a group in Vienna. And with respect to the questions about decoupling symptoms and transmissibility and suppression of the immune response, I think it may be making it more complicated than necessary. I finally found, using the right link to rapidtests.org, a really good diagram that they have there that shows rapid tests versus PCR. But for the purposes of this discussion, I don't know, Vincent, if you can put this figure in the show notes, but there's a gray line that shows the viral load and where it peaks. And then there's a shading of where the virus is transmissible. And in terms of decoupling the onset of symptoms and transmissibility, I think that the gist of it is that the symptoms start to arise around the time when there's peak viral loads, or even shortly thereafter.
Correct. That's
and
right.
So I don't think there's any kind of real decoupling. It's going to be different for every virus, and that's why it's taken us a while to figure out what this timeline looks like.
Yep, exactly.
And I don't think we understand why it's different for different viruses. You know, why in this case is there does the viral load peak before symptom onset on average? Whereas even in something as closely related as SARS-1, that was not the case, right? That you were transmissible and symptomatic at the peak. So I don't think we understand why there are those differences. Certainly, I don't.
No, I agree.
don't either.
I agree. But that the test that is in the preprint, it's interesting. Doesn't seem to require any instrumentation. Uses let's call it a pipette-free version for home testing with open source enzymes. So, I mean, it could be okay, but I do think what do they call it? The lick the strip or somebody.
She called it lick a stick.
Lick a stick.
Yes.
That's what she called it. I like to lick a stick. Lick a stick. All right. Rich, can you take...
Suzanne writes, ProPublica usually does a pretty good job of investigating things, but this really seems like a scaremongering to me. I believe they're well-meaning and they may have a point, but the lack of transparency, about the lack of transparency, but the rest sounds more to me like things are working the way they should. I was wondering what you all thought. And she links to an article in ProPublica that's entitled, "Here Are 6 Accidents UNC Researchers Had with Lab-Created Coronaviruses." And I looked through this. It was a while ago now, but I looked through this, and I think, Suzanne, you've got it absolutely right. Except I'm not sure that they were well-meaning, okay, in publishing this article the way they did. I think it's, some version of clickbait in a way, because it is, it's nuts. There, I don't even think there's an issue with lack of transparency. Okay, things happen in laboratories. There are accidents that happen. Okay, nobody ever said that we weren't ever going to have any accidents. That's why we have high containment laboratories, right? Mm-hmm. So that when there's an accident, it's contained. And this describes some mishaps, by and large, pretty minor, dropping a sample on the floor. Okay, in mostly BSL-3 labs, I think.
Yeah.
Mm-hmm.
And in these, I've never worked in a BSL-3 lab, but my understanding is that there's a standard procedure where there's an accident to not only clean up, but also report it and have the accident reviewed by the appropriate people. And that's all public record. Okay, it's not an issue. Okay, and so this is just routine reporting of lab accidents, and the bottom line is that they're doing their job. Okay, there was an accident. They cleaned up. They did the appropriate reporting and surveillance, and everything's okay, and nobody got hurt.
Yeah.
So this is business as usual.
Exactly. All of these stories then talk about, well, then they reported it, and these are the health things that were done by occupational health, and this seems like exactly what we want people to do when there's an accident, is report it and go through these procedures. And so, yes, this is great.
The headline calling them lab-created coronaviruses. These are mostly all the mouse-adapted coronaviruses. And so, yes, they're lab-created, but that's by passaging these viruses in such a way that they can now grow in the animal model mice. And some of the accidents were mice, escaping and running around on the floor, which
fighting people,
whatever. And again, in terms of, reporting everything that happens, I know of a case where, for these breathing apparatuses, there's a big battery pack. And in one case, the person was in the process of doffing their PPE, taking off all their PPE, and the battery pack fell on the floor of the lab, and they reported that.
Yeah.
So,
that's right.
This is just what is supposed to happen. So I agree with the Suzanne that things here are working the way they should.
It's fear-mongering. It's true.
And I appreciate actually this letter to give an opportunity to point out how what is really business as usual can be misrepresented as some horrible thing going on.
We had Ron Fouchier on TWiV at an ASV. I think it was Fort Collins. And he said, if our computer crashes in the BSL-3, we have to report it. We have to report everything. So the fact that there are reports should not make you think that things are going wrong. It's just what we have to do. Okay. Let's see. Where are we? Paula, right? Everyone's gone. Everyone has gone once, right?
Yes.
I'm a new TWiV listener in Minnesota, USA. Currently sunny, 67 F, 19.4 C where I am. Humidity 66% with calm winds. I am a 50-year-old stay-at-home mom who started college at Michigan Tech ages ago, majoring in med tech with an eye toward vet school before getting married and working to get my husband through his school. I did not finish my degree. Yes, we are still married 29 years this coming November and raised 3 wonderful kids and have 2 sweet grandsons now. Regarding the sudden spike in New Zealand cases, many are saying that the virus isn't likely to be transmitted on surfaces, including frozen food, which they think might be the source for these new cases, but I haven't found any story that mentioned anything about the phenomena of sublimation of the frost crystals on the surface of frozen items. If you're handling frozen food and some of it is contaminated with coronavirus from other workers breathing, coughing, or sneezing on it, then every time air passes over those packages, especially as they pass into a slightly warmer environment during shipment, you get some sublimation, and you are likely to inhale some of the sublimated vapor, wouldn't you think? Thanks for helping to oil my rusty brain. Lots of the discussion is going over my head, but it sure makes me feel more intelligent. Sublimation.
So I think of sublimation as basically, well, okay, so this is my lab stuff creeping in. I think of it like lyophilization or freeze drying, right? And in that case, if we're going to get technical about this, I don't think the New Zealanders got their virus from frozen food. But from a strictly technical point of view, I would figure that the vapor coming off of some sort of mixture of stuff frozen on a surface would be water and would leave most of the other material behind. Okay. I have this vivid memory of the first time I ever saw something lyophilized. It was a solution. It was in Harry Noller's lab, and it was a solution of ribosomes. Okay. So it's a clear fluid, and he stuck it in on the lyophilizer, and it's in this round glass thing, and
X tube.
you freeze it, and you connect it to the vacuum, and all of the water comes off, and there was this lacy stuff left behind. That's all the protein and RNA from the ribosomes. That's the residue, okay? That stuff doesn't evaporate. Sublimation, I think, is the same thing. It's like a distillation in a way. I think you lose the vapor. I don't think you lose the material. So that's my spin.
Right. I agree. I don't think that the virus on frozen food is the cause of the spike in New Zealand cases. No. But I did look up sublimate, and it is to be transformed directly from solid to gas,
right,
or from gas to solid without being liquid. So I wasn't aware that it could refer to going from gas back to solid. But
so if you go from solid to gas, let's say you go from ice to gas, and there's some coronavirus on the ice, it's as Rich said, it's going to remain behind. The gas will not have coronavirus in it, correct? Or is that wrong assumption?
I think that seems to make sense.
Right. Anyway,
when my snowman sublimates, it leaves his eyes and his carrot nose behind.
How many of those do you have in Austin?
Not a lot, but I've been lusting after Sun River and thinking about it.
I think last week we got a letter explaining the New Zealand outbreak where people coming in and the quarantine wasn't done right and so forth. We talked all about that. It was not frozen food for sure. All right. Brianne.
John writes, Dear Twivers, as a Jesuit priest and a huge Doctor Who fan, I felt the need to write in response to episode 654. I teach parasitology at Creighton University and have been an avid listener of TWiP, which I recommend to all of your listeners. Although I have listened to a few TWiV episodes sporadically in the past, I have listened to all of this year's episodes and have thoroughly enjoyed them. Episode 540 brought me a great deal of hope, and so I want to thank you for countering a miasma of desolation. I also want to thank Daniel Griffin, who not only has good taste in sci-fi, but generally gives his time to share critical first-hand clinical knowledge on COVID-19. I also love his case studies on TWiP and use them in my class. Until we get this pandemic under control, stay grumpy and keep up the great work. Sincerely, John, who is in Omaha, Nebraska, where it has been sunny and in the mid-80s.
And I think he referred to episode 640.
I think so too.
Which is,
yes,
the Michael Mina episode.
Gosh, what is 540?
It was pre-coronavirus.
Was there such a time? Wascally viruses.
Yeah.
Wow, long time ago. Yes, I recognize you from TWiP for sending in letters. Who's next? Brianne? Brianne just read that. Kathy.
All right. Dear Vincent and other TWiV hosts, I'm sure you've seen this article by now, but since it so clearly and persuasively echoes what TWiV has advocated since the Michael Mina episode, TWiV 640, I couldn't resist sending along a copy. Keep TWiVing. Regards, Earl. And it's the Atlantic article that I think I pointed out to you because I had read ahead in the listener email and found this one. So I mentioned it last week. It's a really good one.
Yeah, and it's nice that they're making them available outside of the paywall, right? Which is normally, I never read the Atlantic because I don't have a subscription, but it's a good article. Yes, very good. Rich.
Oh, and they have a term in there that I really like, which is the contagiousness test. That these rapid tests should be thought of as contagiousness tests.
Yes, exactly. Yep. Yes, that's absolutely right. Yep. All right, Rich.
Maria writes, Dear TWiV team, I am a quality assurance lead for a diagnostics company. I learned about TWiV from a grad professor of mine and want to thank you all for keeping me sane and informed during this crazy time. A couple of weeks ago, my 17-year-old nephew started feeling ill and soon thereafter lost his sense of taste and smell. He had access to an Abbott ID test and his results came back positive. He has spent the summer working in a restaurant and 3 of his co-workers were also diagnosed positive soon thereafter. All of his friends and family who have had exposure to him have since been tested using lab PCR. All of their tests came back negative, including those of his immediate family and of his girlfriend, who he was swapping spit with the day before his positive test result. Do you have any explanation for this? My brother is now convinced that there is a, quote, different strain, unquote, of SARS-CoV-2 in our hometown that is less transmissible. I ineloquently told him that he was wrong, but couldn't explain how my nephew did not infect any of his friends and family. Keep up the excellent work. My gratitude is expressed in the following haiku. And here's the haiku. So many false facts, nowhere to turn, so confused. Then TWiV cleared the fog. Excellent.
Mm-hmm.
I can't necessarily explain this phenomena except that it is an anecdote of one person's experience in a group of people, and so it is a data point. Transmission isn't necessarily 100%.
Right.
Just because you've got virus and you're shedding virus doesn't mean that everybody you're going to be in contact with gets infected, even if you're swapping spit with him. Who knows? Maybe his girlfriend, first of all, maybe he was swapping spit when he was down on the downside of his infection. Maybe he didn't have a real high titer.
Yeah.
Maybe she had just gotten over a cold, or in the recent past has been infected with all 4 human coronaviruses and has a little T cell background or something. I don't know. Okay.
Wildly speculating there.
I'm wildly
Yes.
speculating, you bet. Okay, but I don't think it's a big issue, and certainly the notion that there's a different strain is nonsense.
Yeah,
right.
Absolutely.
The word that popped into my mind was stochastic, and then
Yes.
I was thinking, well, that's maybe a little bit beyond some people's vocabulary, and you can just use the word random.
Yeah.
So it's
Mhm.
random that the other people around him did not become infected.
This reminds me of some correspondence I had with. I now get independent correspondence with TWiV listeners who, send me stuff. Fine, okay, and this was about, I guess. What is we had this discussion. What's the infectious dose of a virus?
Mm-hmm.
And, I forget what the vocabulary was, because there was what's the infectivity or something. And you guys said it's 1. Okay. Any given particle or any given active particle is potentially infectious, but the infectious dose is a different story. Okay. On average, how many infectious viruses does it take to infect somebody? And in fact, the paper that this person enlightened me to said that the measurements had been done for SARS-1, and I think I kind of forget, but there was some at least some modeling for SARS-2, and it's around. They came up with the ID50. On average, the dose required to infect 50% of people who were in an encounter was about 250, 280 infectious particles.
Mm.
Okay. So it's not 1. Okay. And that's a statistical measure. Sometimes maybe you get 500 or 1,000, you don't get sick.
I just thought of another example, and that is one that really befuddled some faculty colleague of mine. If we're working with genetically identical mice and we infect them all with the same dose of virus, why don't they all behave the same way with respect to how sick they get, or do they die? And we just had to tell them it's biology. It's random. It's just the way it is.
It reminds me of a phrase, the biological correlate of Murphy's Law. Under carefully controlled environmental conditions, the organism does what it pleases.
That's good.
Oh, that's another good Rich Condit quote.
I like that.
Write that
I like
down.
that.
Yes.
Right.
A friend of mine mentioned that perhaps if we put together all of these excellent TWiV haikus and limericks and other poetry, we could call it TWiV verse.
That's good.
That is good.
Well, as you note here, I have a note saying I should set up a page of haiku, limericks, poetry.
As if you didn't have enough to do.
Yeah, right.
But you need to somehow highlight these things or something so that you can get back to them or harvest them in real time. This is a good one. I like this one.
Okay, Ken writes, sends us a UCSF press release. Makes this sound like potentially a real game changer. It references this preprint. Would be great to hear what the TWiV team thinks. P.S. Rainy 17C near Oxford, England. Not unusual. So this came across my radar a couple of weeks ago, and they call this aeronabs. So what did they do at UCSF? They identified single-chain antibodies against the virus that neutralize infectivity, and they identified a bunch, and some of them are very potent. Okay. But the twist here is that they say, why don't we spray this into people's noses and prevent infection? And the only experiment in the preprint that they do to address that is to show that if you make an aerosol out of these monoclonal antibodies, that they still block infectivity. So there's no experiment done in an animal, although I think there are some ongoing to address this. So it's possible that it might work. I mean, I would think you'd probably have to spray it in your nose pretty frequently, and I'm not sure that would be a good thing to be spraying antibodies in your nose on a daily basis. We'd have to find out in a phase 1. Well, we'd have to do a lot of preclinical studies first, of course. But, the idea of spraying monoclonals into your nose and then you'd be protected against infection. I don't know. Is that a good idea?
Yeah. I read this. I actually got interested in this paper, and I read it pretty closely because the technology is quite interesting. There are these that, as I understand it, Brianne, maybe you know more about this than I do, and you can correct me. But as I understand it, they start off with a yeast library where individual yeast in the library express what are basically binding domains from single chain antibodies. They aren't even the whole single chain antibody. They're binding domains. And they can then use an antigen like spike in this case to select out of that library yeast cells that express a binding domain that bind spike. And these are, like 150 or 200 amino acids long. And so they screen a bunch of these, and they get some that bind the spike in a particular way that is neutralizing, and they even have structural data that shows how it binds. And then they do two things. One, they do mutagenesis on them and select amongst the mutagenized guys for guys that do that job better. And the other thing they do, prompted by their structural data, is to multimerize them. So they come up with a mutation and affinity matured antibody multimer that is really good. And among other things, these generally have the characteristic that they're very stable, which is why you can aerosolize them or heat them up or whatever, and they remain stable. The idea of using this as a nasal spray, aside from the fact that they're stable, is discussed in one sentence in the discussion that has a reference to a paper from 2015 where somebody used a monoclonal antibody to respiratory syncytial virus and sprayed it in the nose of cotton rats and showed that it helped diminish or prevent lung infection with RSV in that animal virus. One paper, different virus, animal models. I'm not buying stock in this yet.
Right. Okay. Brianne, you're next.
All right. Lori writes, I'm a pediatrician in San Francisco, and I received an MPH in epidemiology at UC Berkeley. My sister, who is a microbiologist, turned me on to TWiV, and I started with the Michael Minute episode, and I was an immediate convert. I have told anyone and everyone, from colleagues to friends to patient families, to watch it, and I have been shocked that this has not hit the front page of every major newspaper. However, over the last week, I have heard more references to it on the radio and from colleagues. Michael Minna was featured on the UCSF town hall last week, and that created a local buzz. I think this is starting to happen. Please do not stop. Do not discourage. You guys are amazing. You are going to change the world. Thank you. I would love to do something to move the quick spit test forward. I have 2 sons who are in college, and this is no way to learn. They need to get back into the classroom, but of course they also need to be safe. Please let me know if there is anything me or my colleagues can do to fan this fire. Thanks again. I am such a fan. All right. Thanks, Lori.
I'm glad it's getting momentum.
I want to very briefly revisit that previous thing and make the note that I really like the paper from UCSF, and the point of the paper is not that they're making some sort of vaccine. As a matter of fact, it just describes the antibody. It's titled "An Ultra-High Affinity Synthetic Nanobody Blocks SARS-CoV-2 Infection by Locking Spike into an Inactive Conformation," which is absolutely true, with 5 or 6 equally contributing first authors, Schuth, Faust, Sanders, Sangwan, and Rezeljed. I can't pronounce that properly, with the senior author Manglik from UCSF. It's a great paper. I think these antibodies could have a lot of utility for various purposes. I think of them as putting them on, what are we calling it? Lick a stick.
Mm-hmm.
They might be great for that. Okay, because they're really stable.
Could be.
And they bind really hard. Yeah. So I think this technology is fantastic. And these guys, it's the press release that's twisting this into some amazing therapy.
Yeah, it's totally the press release. The press picked this up in multiple places, basically on a nose spray, which, as you say, is not really the focus.
Right. That's not the point
of the paper. But how would they know otherwise to interpret it, right?
One other thing that Lori mentions in her email is about something on the Rockefeller Foundation site where she saw a background photograph where a person is wearing their mask below their nose. And I see a cartoon type of diagram there where that's the case, but I'm not sure if that's what she's referring to, but basically wanted us to call it out, and maybe the Rockefeller Foundation could fix their website to not show improper mask wearing.
Yep. It's ridiculous. Kathy, can you take the next one?
Lizzie writes, "Dear TWiV, in episode 654, you read my email about convincing friends and family not to tell their Congress critters to buy the Russian vaccine. An important part of my question, however, was how to communicate that vaccines coming out of Operation Warp Speed will be fully tested for safety and efficacy by spending money and resources instead of time. Fast, expensive, safe, and effective. The Russians did a combined phase 1-2 trial and declared victory, fast being the only shared virtue. How do I explain the difference without putting people off vaccines altogether?" And then I'm not sure who had this parenthetical remark. 74 patients is not a proper phase 1-2 trial. Oh, that's from Vincent. Okay. So I think that you just have to keep saying that our system, based on past experience, will give us a safe, efficacious vaccine. And working fast by spending more money and more resources is not to the detriment, hopefully, of the resulting vaccines.
No, the numbers alone, the phase 1-2 of the Russians, 74 patients, is ridiculous. We are doing properly populated phase 1, 2, and 3 trials. It's just that we're accelerating it. As Tony Fauci said to us, the only thing we're going to lose is money. We're doing everything the same way. We're just accelerating it. So that's the key. The Russians didn't even really do a proper phase 1 trial.
And I don't know if you all have seen this, but have you seen any of the data from that? Because I think that the fact that, we're seeing data and there's some transparency on our trials, of course, is another big difference.
No, I haven't seen anything.
I think one thing that Lizzie could do to convince her friends is to remind them of the thalidomide era, where countries outside the U.S. approved this drug, and the FDA refused to approve it because it didn't meet their criteria. And it turned out to be, when taken in pregnant women, have devastating consequences. And so the U.S., historically at least, is capable of making judgments on these things that are different than other countries and turn out to be correct judgments. And I have no reason to believe, or very little reason to believe, that at this point in time it will be any different. And in fact, this whole thing with this whole dust up recently with the convalescent serum is kind of a warning shot in that regard. Okay. And I hope people are listening.
Rich, can you take the next one? Daniel
writes, I've started following your podcast after hearing about episode 640 with Michael Mina. That really got a lot of listeners. I wanted to get your thoughts on COVID transmission among young children and implications for sending them back to school. We have a 5-year-old and are contemplating sending him to a school that only has very young children, ages 2 to 6. The school will be practicing social distancing and mask wearing, hand washing, and holding 75% of the class time outside. Class size is limited to 13 kids and 2 teachers. We had felt pretty good about sending our child there in light of news we heard about studies that young children under 10 are much less likely to catch and spread the virus. So those are 2 different things there, catch and spread, okay, that need to be segregated. That's my own little editorial. We'll come back to it. That children under 10 are much less likely to catch and spread the virus, although we understand that can still happen. And when they do become infected, have much milder symptoms. We were particularly encouraged by news regarding a study from South Korea showing much lower rates of infection in younger children. But Sanjay Gupta on CNN today pointed out that the South Korean study only had a very small cohort in the under age 10 group. So he said he was disregarding his study and keeping his kids home from school. Although I found his study, and she gives links to both of these. Although I found this study from Iceland, gives another link, which used larger samples of children under 10 and found much lower rates of infection than in the rest of the population. I'm a trial attorney, and while I'm accustomed to diving into new complex areas to learn them quickly, I don't have a scientific background, and it's a bit difficult to decipher the various incomplete data out there. So my questions are, do you believe that there is a substantially lower infection and transmission rates among young children? Number 1. Number 2, given the school setup described in my email, would you send your child to school? I should add that I presume this calculus will change if the Michael Mina type testing is made available. I saw over the weekend that Yale and the NBA have developed something similar, and I've already made inquiries to see if the school can use those on staff and kids. Thanks for all your work, Dan. So, first of all, once again, I want to emphasize over and over and over again that catching and spreading the virus are not, They say this. The sentence was that young children are much less likely to catch and spread the virus. I see. No, those aren't necessarily two different things. I misspoke. Okay. If you catch the virus and there's no evidence that children spread it any differently than anybody else. So my answer to question number one: do you believe there is a substantially lower infection and transmission rates among young children? No, I do not believe that. Okay, I've not seen any data that can, although I'm not an expert at this, I've seen nothing that convinces me that kids are infected or transmit any less. And I think there is a lot of confusion out there based on the fact that children are less susceptible to, when they are infected, they less frequently get disease. They seem to have milder disease, more asymptomatic infections. That does not mean that there's a lower infection rate. It does not mean that they transmit any less. So unless I see some huge study that convinces me otherwise, my assumption is that they get infected, because everything I've seen said that there's a lot of infection in children. We did one paper that says they cook up even more virus in kids less than 5. There are not a lot of transmission studies, but there's no reason to believe that if you're cooking up that much virus, you aren't transmitting it. So I would treat kids exactly the same way as adults. Would I send my child to that school? I don't know. I'd really want them to have a testing program in place. It's really, I don't worry about the kids so much, because they don't get sick very much. What I worry about is the transmission. If they go to school and get sick, they're going to bring it home to me. How do I feel about me? They're going to spread it around. They're going to spread it around to the rest of their contacts. Okay. Are their grandparents involved? To what extent are you going to contact trace your kids and quarantine them? These are all factors in that decision.
I think also the rate of positivity in the community is important.
Yes.
Right? If it's high, forget it. If it's over 1% or 2%, no. But if it's less than 1%, then it bodes better, at least for not triggering another outbreak.
Then contact tracing is going to be much more possible as well.
Yeah, that's right. But I would really like to have testing a part of it. I think that's really important. Brianne, can you take the next one?
Sure. Steve writes, thanks for being maybe the voice of reason in these chaotic times. I am an internist and hospitalist caring for COVID-19 patients on a regular basis. Your podcast has been a great source of information and reasonable opinion for me and my group. I suspect this question asks for an opinion because I think that it is something that cannot be known yet. To what degree do you believe the improvements in morbidity and mortality among COVID-19 patients can be attributed to the natural selection of the virus towards a less lethal phenotype versus the changes in how we are managing them medically? Is what we are doing medically really helping the patients that much, or are we just watching the natural course of a viral pandemic as it evolves? Thanks for any insights you can offer, Steve. I would say, Steve, that we have not seen any evidence that we are seeing a lot of change in this virus or natural selection towards a less lethal phenotype. I think that it really probably is about what you guys are doing managing them medically. I would lean much more towards the medical management than anything about natural selection.
I agree. And I think Daniel's very interesting on this topic. I'd love to hear him talk about this. And it makes me think, I was going to comment on this last time, but it makes me think about procedures in a laboratory. Okay. I think I've commented on this before. If you have a procedure that you do in a laboratory that, you do a series of steps that leads to an experiment, typically first couple of times you do it, the outcome, it's just messy. It's awful. Okay. And if you're paying attention, you do it over and over and over again. It gets better. Okay. If you're paying attention, and you never know why. You look back and you say, what am I doing differently? And you don't know. Okay. But you're probably doing lots of things differently because you're paying attention. Okay. And I get the impression from Daniel that while there are some obvious things that they're doing differently in how they're managing the patients, that I have to wonder if there aren't a lot of subtle things that go on that just improves the patient management.
I think if you were to go back and listen to some of the early TWiVs when Daniel came on and gave his clinical reports about what we knew and what they were doing, and just the one from last week where he again outlined, what happens each week and how we know so much more about it and what are the things to do as a clinician at each of those different stages. Definitely improvements in the medical treatment. And as Brianne said, no evidence toward selection of a virus with a less lethal phenotype.
In particular, Daniel talks about throwing the kitchen sink at him. I have a feeling that, when you're presented with this kind of stuff, I get the impression that as a physician, when you're presented with this kind of stuff, you try everything you got. And over time, you find out that many of those things were doing more harm than good. A ventilator is a good example of that. You put that off as long as you possibly can because people apparently crash when you put them on a ventilator. In the end, you don't, I guess, have any choice, but you put it off as long as possible.
Kathy, can you take the next one?
Sure. Talia writes, Dear TWiV team, hello from Berlin, Germany, where things feel relatively normal. In fact, if you go to the lakes and beer gardens, which are packed, you can easily forget there's a global pandemic going on. Groups of people are meeting, people are hugging, going to routine doctor's appointments, and the subways are getting crowded, though everyone wears a mask. The biggest news item recently was a naked bather who chased a wild boar around a lake in Berlin. Yes, there are pictures. And Talia sent the link to The Guardian, and it's hilarious. So you should stop watching or listening now, go to the show notes, find this letter, and watch this video.
Pull over your car. Don't hit an embankment.
No embankments. Okay. But I think everyone is bracing themselves for the fall, so it feels a bit like the calm before the storm. Infection numbers have been on the rise for the past few weeks. Yesterday, with over 1,700 cases registered in the past 24 hours, was the highest it has been in Germany since the end of April. My husband and I traveled here from Newark, New Jersey in early June. It was not the most pleasant experience. We wore our N95 masks for 15 hours straight, and our flight from Newark to Frankfurt was completely full. But somehow we survived without getting infected and are now happily drinking beer in beer gardens and dodging wild boars. I don't have any sophisticated virus questions, but I did have 2 puns I wanted to share with you. It came to me the other day in a rare moment of inspiration when I was listening to your interview with Adam Kucharski. I think they would make fun logos for TWiV merchandise, maybe t-shirts. Viruses are zero. Oh, it should be viruses are not alive.
Yes.
So read the zero as a not. Viruses are not alive, or viruses are naughty. All the best from Germany, where it is currently 78 degrees Fahrenheit. Thalia.
Wow. 15 hours. Wow.
Yeah.
That's a long time.
And this is the Thalia who did that wonderful
it is.
podcast of her own on TWiV. Yes. It's really great. Yes.
Well, next week we will have, is it next week or the week after, Christian Drosten to give us a German update.
Good.
Das Coronavirus. Is it next week? Because that's your little mini holiday.
Mini vacation,
Because when I hear that in various locations they're going back to business as usual, I go, I'm not so sure about that. Though, it could be that if everybody is actually maintaining a social distance and everybody is wearing a mask, maybe that has a very, very significant mitigating effect. I don't know. Let
me just give you some numbers from a Washington Post article yesterday. COVID-19 deaths reported the previous day. Italy 3, France 9, Japan 14, Canada 7, UK 18, Germany 3, United States 974. So all those
we're crushing this.
Yeah, we're crushing.
We got it all under control.
Disaster. All right, last one from MJ. Don had recently asked about the need for many to avoid complying with any reasonable actions or standard of care in the address of protecting others from infection now with COVID-19 and presumably for any other bad infection or pandemic. If I may suggest, the old Freudian defense mechanism of denial is the best solution to Don's query. The inability to face some bad past event or some potential harm or one's own potential responsibility or the potential for such responsibility is met by the inability to either acknowledge the risk or the situation behind the risk itself. Yes. So this was a letter who someone wrote, I don't understand how people can deny these things and do these things in the face of what's going on. In a manner analogous to Holocaust denial that COVID-19 can exist as severe, the business is no more than a simple case of flu, or that it exists, Trump's use of it's a democratic hoax, or that it is transient, Trump's use here of it will just go away, or the lack of need to respond to it as the person speaking believes that he or she or they could not harbor the virus and would not likely transmit it due to a lack of contact or lack of symptoms or both. All of these are patently false, but the need to maintain one's own ego gratification while acting in a possibly juvenile and self-centered manner overrides any concept of responsibility. All of this jives with Freud's mechanism of a person is faced with a fact that is too uncomfortable to accept and rejects it instead, insisting that it is not true despite what may be overwhelming evidence. And then provides a link to an article called Defenses, November 3rd, page construction, currently rewritten by Richard Nyolan on April 8th, 2011, but documented on another page. Keep up your wonderful work. Well, thank you. I always like hearing explanations for these blatant ignoring of science, right? And it's denial. It has its roots.
This is one of the reasons that I enjoy doing this with you guys so much, and probably the reason that people like listening to it, because I feel like I'm constantly awash in, the news, which is just insane. And this is an island of sanity and all of that.
I don't mean to politicize this outbreak. I try not to. However, the other day, was it CBS News? Yes, did a survey of 2,000 registered voters. And they asked, has the number of U.S. deaths from coronavirus been acceptable or unacceptable? 57% of Republicans said the number of deaths was acceptable. 10% of Democrats said the number of deaths was acceptable, and 33% of independents. So tell me, in what world does it matter what party you ascribe yourself to decide that a death of any kind is acceptable or not? I just don't understand it. Maybe I need more explanations from MJ that I'm not getting this.
Yeah, I think MJ has some insights into this. There's a kind of a denialism involved.
Yeah.
But I mean, just to say that a death is acceptable on top of it is crazy, right? I mean, it's crazy. No death is acceptable. Human beings are amazing. Well, they can be.
Oh, even when they aren't, they are.
Actually, what I mean is biologically and, as an organism, they're amazing, right?
Well, even crazy is amazing, right?
I suppose it's all the human condition, right? Well, it's getting twisted now. I don't want to go any further. Let's say that is TWiV 657, and I am just amazed that we are 57 episodes past number 600. Wow, it went fast. Microbe.tv/TWiV is where you will find the show notes. I have the unusual situation of having an insect buzzing around, which I never do in my office at Columbia. Microbe.tv/TWiV. Send your comments, questions to TWiV at microbe.tv. If you like what we do, consider supporting us. Microbe.tv/contribute. Brianne Barker is at Drew University. On Twitter, she is BioProf Barker. Thank you, Brianne.
Thanks. It was great to be here.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun. And last time I did wait until the end and I heard the announcement about chat with a virologist, the thing that you can do by going to asv.org, scroll down, and you'll find the link to chat with a virologist. If you want to have someone come to your educational Zoom or whatever type of meeting and talk to you about viruses.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure. And thank you. Thank you, Brianne, Kathy, listeners. This is an island of sanity for me. I enjoy it.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.